Good afternoon, everyone. This is Ernie Bush from CHA, and on behalf of the Drug Safety Executive Council, I welcome you to today's webinar. Today we're going to explore a new initiative called CiPA that is intended to provide a more rigorous and, shall we say, modern or rationalized approach to arrhythmia prediction for preclinical studies. We have two presenters today, and I'm very happy to introduce them. The first presenter will be Dr. Philip Sager. Dr. Sager is a cardiologist who has held senior leadership positions within the pharmaceutical industry and academia and has played a significant role in innovative drug development as well as the assessment of cardiovascular safety issues in the pharmaceutical development. He has played key roles in the development of Zetia, Vytorin, Crestor, Letairis, and Brilinta. He was the ICH Expert Working Group RMA topic leader on the QT interval initiative, that is E14, and is the chair of the Scientific Programs Committee and member of the Executive Committee of the Cardiac Safety Research Consortium. Thank you, Doctor, for being available, Dr. Sager, and we look forward to your presentation. And our second presenter will be Dr. Arthur or Buzz Brown. He is the ChanTest founder and president and CEO and an adjunct professor of physiology and biophysics at Case Western Reserve University School of Medicine. Arthur or Buzz is a trained cardiologist and physiologist with more than 30 years of experience in ion channels and their relationship to human disease, and has published extensively in peer-reviewed journals. Dr. Brown has established world-leading physiology departments at the University of Texas, Medical Branch, Baylor College of Medicine, and Case Western Reserve. And I think I would add that ChanTest is by far the gold standard for providing cardiovascular safety evaluation services. So without further ado, I would like to turn it over to you, Dr. Sager. You've been passed the ball, and you can advance the slides with your presentation.
Advances in pro-arrhythmia assessment, and a very exciting new approach, which is to focus on non-clinical ways of evaluating pro-arrhythmic risk and really moving a lot of this from more the clinical area with thorough QT studies to the preclinical area with a lot of potential benefits. And hopefully this new paradigm would let us move forward and have new ways of evaluating pro-arrhythmic risk without the thorough QT study. So when we think about Torsades de Pointes, it's been the single most common cause of withdrawal or restrictions of marketed drugs prior to the ICH E14 guidance. And that really resulted in the decision, the 2001 timeframe, to go ahead and create a clinical guidance. And one of the conundrums is that torsade is very rarely ever observed during clinical development, except for drugs that have a high pro-arrhythmic potential, such as some of the class 3 antiarrhythmic agents. And thus there was a need for what to evaluate, what surrogates to use, since torsade is very rarely ever seen during the clinical development stage. So the focus was on hERG testing as well as the QTc interval. And turning to the QTc interval, it's sensitive, but it's not very specific. And particularly having around a 5 millisecond boundary for a negative thorough QT study has resulted in many kind of mildly positive thorough QT studies, which has impacted both labeling but also drug development. And shown here at the bottom of the slide is just a typical episode of Torsades de Pointes, which stands for twisting of the points. And if you look at the major deflections of the QRS complexes, they go from negative to negative again, and then to positive, typical for torsade. And these arrhythmias are typically very fast, and should they continue and become sustained, people usually lose consciousness, and thus they can be mortal. In other patients, they do stop prematurely and may result in lightheadedness or syncope. So in doing a thorough QT study, it's become clear that there are really consequences that are pretty significant of developing a drug that has a significant effect on the QT interval. In ICHE14, the upper bound of having a positive, that denotes a positive QT study, is a one-sided 95% confidence interval of the QTc increase placebo-corrected greater than 10 milliseconds, which results in about a mean increase of 5 or 6 milliseconds. And the consequences of developing a compound that has a significant QT effect includes a significant development burden. Increased perceived risk results in the need to potentially have a greater degree of benefit, so the overall benefit-risk relationship remains positive. Increased costs, there can be significant amounts of additional testing that needs to be done, potential for delays in filing or approval, label warnings, and also competitive implications. If one develops the first novel drug for a certain disorder, and then a year or two later, someone else develops a drug also for that disorder that doesn't affect the QT interval and doesn't have label warnings, that can have significant competitive and commercial implications. Licensing and partnering issues, one of the first questions that people ask if they're looking at potentially in-licensing a drug is what its effects are on the QTc interval. And because of these reasons, showing an effect on the QTc interval often leads to terminating drug development, and sometimes, as we'll discuss, this can be premature development because not all QTc increases actually denote proarrhythmic risk. Again, as we said, the sensitivity is high, but the specificity is not. So ICHE14, as well as that should be S7B, have really had a lot of impact since these were put into effect around 2005, and they've really resulted in no drugs that had unrecognized risk being approved by regulatory authorities. So we've moved from a time period in 2001 and beforehand where this was the most common reason for drugs to get restrictive labeling or to be removed from the market to a time now when we really have not put new drugs on the market whose risks we didn't understand. So that sounds really successful. So what are we talking about today? Why would we even think about doing something different? Well, the reasons is there's also been intentional negative impact on drug development. The focus on hERG as well as QT has led to premature discontinuation of drugs that might have really had a significant benefit to public health. In some of these drugs, an inaccurate perception of risk and the development burden of continuing to develop them, the cost of doing that, the labeling implications, has resulted in drugs being terminated from development that really could have been beneficial. And this has been a particular concern in certain areas because of cross-reactivity with some of the cardiac ionic currents, such as in the CNS area. And many of the drugs currently that have QT labeling are probably unlikely to be truly proarrhythmic. So if we look at the different ion channels responsible for the ECG QRS complex on the left-hand side, or the action potential duration below that on the left, there are many different ionic currents, and not all of them are displayed on this slide. The one that we normally think of as being playing the largest role in the development of Torsades and QT prolongation is the delayed rectifier potassium current, which is measured by hERG. But there actually are many other currents that are responsible for action potential prolongation. And those are shown right here. Not only there are a number of currents, but modulation of one current could potentially reduce the proarrhythmic effects of QT prolongation of another current. So, for example, with hERG blockade, if one impacts the late inward sodium current, you still may have QT prolongation, but not actually have a risk of Torsades. So again, another way to dissociate QT prolongation from Torsades is by affecting potentially multiple currents and just looking at hERG alone. won't give you the whole picture in terms of proarrhythmic risk, since, for example, IKS, the slowly activating component of the delayed rectifier potassium current, is another way to cause torsade. So a hERG focus doesn't tell the whole story either. So what are some examples? Sodium pentobarbital prolongs the QT interval but does not cause torsade. It inhibits multiple ionic currents. Amiodarone, torsade is very rare. It does occur, but you have to think of this drug often can prolong the QTc interval to 500, 550, 580 milliseconds. So marked QT prolongation, torsade is very rare. Verapamil has a balance between blocking IKR as well as the calcium, L-type calcium current. And QTc is usually not prolonged except with very high exposures, but again, not associated with torsade. And ranolazine also prolongs the QTc interval, but not associated with torsade. So when we look at these, what we see is these drugs in preclinical assays do not cause early after depolarizations. They tend to reduce dispersion, and they impact multiple ionic currents that, despite the QTc prolongation, these drugs are safe from a proarrhythmic standpoint. So the issues we face today is the QT prolongation does not necessarily result in proarrhythmia. hERG block also does not necessarily result in proarrhythmia. There's been a negative impact on drug development. This finding of what might be a hERG signal that can result in QTc prolongation or actual QTc prolongation often leads to termination of drugs that might really be beneficial. And thus the benefits of a new paradigm where we take our current mechanistic understanding of torsade and apply it so we don't measure a surrogate, but we actually measure the true potential of a drug to be proarrhythmic, could have multiple benefits on drug development. So this would focus on the real issue, which is proarrhythmia. It can result in reducing the premature termination of drugs with favorable benefit to risk profiles. Also make drug development more efficient. It would move the bulk of the proarrhythmic assessment from the development stage, where it often is now, to the discovery period. And thus, before one even gets into the expense of clinical testing, one would be able to exclude drugs where it was thought that there was a significant proarrhythmic risk and the benefit to risk ratio wouldn't be positive. One could also potentially use the assays to guide candidate selection. So one could use them to decide which candidates to move forward. And obviating the need for the thorough QT study also would be a significant cost savings, as well as, again, would be part of moving the focus to the non-clinical discovery period. In fact, there could be some really significant benefits early on of such an approach, and that is the accuracy with which drugs are currently labeled could be improved, and this could actually result in relabeling of some of the drugs that are currently on the market. And there are drugs that aren't used as much as maybe they should be because of concerns about proarrhythmia that may not be actual real risk. So there was a meeting held last summer at the FDA that discussed this in detail, and this is an article that actually says in press but actually has been published in January 2014. And this discussed a new paradigm. And the concept here is that proarrhythmic risk could be determined by preclinical assessments. One would examine the proclivity to develop early after depolarizations, and one would do this through in silico modeling modeling the effects of a drug on a number of ionic currents as well as a cell-based approach. And this would focus on high throughput types of approaches for proarrhythmic assessment, as well as also evaluating the ECGs in phase 1 in order to make sure that what was seen through the non-clinical assays, that there are no surprises when one gets into humans. So I'll now discuss this over the next few slides. And shown here is and early after depolarization. And what's really changed over the last 10 years is we've developed a much deeper understanding of the mechanism of torsade de points. And this lets us really move from using surrogates to actually directly measuring prorhythmic risk. So we know that the prorhythmic vulnerability is linked to impairment of repolarization, but also critically instability in the resting membrane potential during phase 3. And this can result in early after depolarizations that can result in triggered activity and the genesis of torsade de points. And the ease of inducing EADs reflects prorhythmic vulnerability. So the concept here is it wouldn't be kind of a positive assay and negative assay. So that's how the thorough QT study is looked at. It's either positive or negative. But actually one could create a gradation of risk from extremely low risk to much higher risk of torsade. And so moving from a binary approach to really a gradient of risk approach, because for some drugs, depending on their use, let's say in acute myelogenous leukemia, one's willing to accept certain degrees of risk. Or obviously for allergic rhinitis, that wouldn't be the case. So it's important to be able to assess drugs across a gradient. So there are two major non-clinical pieces to the CiPA approach, and that is to examine ionic currents and to use these ionic currents in an in silico model to be able to model a prorhythmic potential, so a sophisticated computerized model. So one would examine multiple ionic currents using voltage clamp studies and then run them through this model. And then the results of that could be verified in a myocyte-based approach using human ventricular myocytes. And again, there would also be assessments in the phase 1 clinical ECG evaluations, making sure there was nothing unexpected. So these are be complementary approaches, and they're not designed to actually see whether or not an arrhythmia is induced, but rather to look at a prorhythmic potential. So one important piece of this is determining which ionic currents need to be assessed and developing standardized approaches to doing that. So this would be voltage clamp studies, probably focused on the 6 currents that I showed you earlier, looking at the effects on these currents using heterologous expression systems of human channels. And right now there's efforts going on to establish the best practices, standardization, as well as choosing exactly which currents need to be examined and which properties. It's probably more than just the IC50, but rather over a range, and whether or not things like rate dependency also have to be evaluated. This would use a higher throughput automated patch platforms. This data would then not be looked at individually, but would all feed into an in silico model of a reconstruction of the action potential. So this is a computerized model that's in the process of being developed, but is likely to be based on the O'Hara and Rudy model. And this examines global effects on repolarization using the multiple ionic currents that are measured in the patch clamp studies. And it's focused on examining the proclivity to develop early after depolarizations as well as instability during phase 3 of the action potential and repolarization. And this is a large effort that's currently underway, and I'll come back to that later. The other part is using human cardiac myocytes, and this is based on using a stem cell myocyte approach, using well-characterized physiologic recording conditions, and focused on action potentials, repolarization, and the morphology of the action potentials, and really is to, at the moment, is to verify what's seen in the in silico models. But eventually, as cardiac stem cells further develop, and this is an extremely rapidly moving area of science, we expect that over time, this may be able to become the main focus of proarrhythmic assessment. But at the current time, the focus is, again, looking at proarrhythmia here, both verifying the in silico approach as well as looking at other measurements of proarrhythmia in the stem cells. And again, this is also the other piece of this that's important is that the ECG evaluation of humans in phase 1. So by the way, I should have said earlier, CiPA stands for the Comprehensive In Vitro Proarrhythmia Assay. So this can really have we think, a major impact on the efficiency of drug development. So the approach here is to standardize the in vitro patch clamp assays as well as creating and perfecting in silico modeling of drug effects. It's going to define the role of human cardiomyocytes to inform the proarrhythmic potential of drugs. And again, this is a very rapidly moving area of science, and we expect the role of this to increase over time. To provide proarrhythmic ranking based on calibration using agreed-upon standards. So drugs would be ranked from very low to quite high risk instead of the current binary approach. And it's really designed to really significantly change the current status quo and move away from imperfect surrogate markers with the expectation of replacing the thorough QT study. There are a number of work streams that have been put together for this. The in silico modeling, which is being led by Tom Kalatsky at the FDA. The ion channel work, which is being led by several people, including Cyril Pettit at HESI. The stem cell myocyte work, which is being led by Gary Chantant at AbbVie. The compound selection for which compounds will actually be tested and the clinical translation as well as regulatory approaches, which I'm overseeing. And then there's a steering team, which is overall being, which includes these people plus a number of others, including Derek Leichman and Hugo Vargas and representatives also from the EMA and the PDMA in Japan and Health Canada. So there's a wide range of regulatory input and scientific input. So in summary, this is really, I think, a very exciting time in cardiovascular arrhythmia assessment. This is really likely to result in a major change in how we think about assessing proarrhythmic potential. There's the potential to markedly change the current approaches with a very positive impact on drug development. And these work teams that I already showed you are really working hard on this and moving the process forward. And so there's a lot of scientific work, and there's a lot of opportunity still for people to become involved if they'd like to be. And then this concept of using a human cell-based approach with a deep understanding of the mechanisms raises the question about whether this could be extended to also other areas in other types of safety assessments. And that's something that people are thinking about, trying again from other areas also. Maybe in cardiotoxicity, for example, moving to a more mechanistically based approach that doesn't use animal cells, but rather uses human cells. So I'll stop here, and I'll actually got just one more slide here that just shows there are many collaborators that have been working on this. This has been very much, it's a very large group effort. So I'll stop here and turn it over to Buzz. Thank you.
Thank you, Dr. Sager. That was excellent. And already we've received some questions that I have, at least on my system. So we will be having a further discussion on these topics, but I did want to remind everybody, if you have questions or comments from or to the presenters, by all means, put them in the QA module there at the lower right-hand parts of your screen, and and we'll assemble them and present them to the presenters. So with that, I would like to move to the next presenter, Dr. Arthur, or Buzz, as everybody knows him, Brown from ChanTest. Good afternoon, Buzz. At least on my system, there you go, you're muted. You're off mute now, and you have the ball, so go ahead and introduce yourself quickly, Buzz, and then we'll look forward to your presentation.
Okay, thanks, Ernie. Thanks, Philip, and hi, everybody. I'm going to try to make tracks through here so that we can leave time for questions. Some of the material I'm going to present fairly cursorily because Philip has covered some of it anyways. So I'm Buzz Brown. I'm the founder, president, and CEO of ChanTest, and I have an adjunct professorship at Case. Just briefly, I think we've heard about the present guidances. I'd like to present perhaps an idiosyncratic view of their histories, but since I've been involved in, for example, the development of the Hurg assay and its application to cardiac risk assessment from the very beginning, maybe show you where I'm coming from, some of the problems with the present guidances from a practical point of view, and I think we're definitely signed up. We were marching in the direction of CiPA anyways, and I think that has a strong case has been made for moving in this direction. And CiPA is essentially where we're saying that we want to shift the emphasis from QT prolongation to proarrhythmia, and we want to shift the non-clinical side of things and the clinical side of things for that matter. We want to shift that to a more comprehensive and method of action kind of a mechanistic kind of approach. So this slide, the inset there shows where we got involved, which was back in the days of Seldane or terfenadine, which was the first non-sedating antihistamine. It was a blockbuster drug, and it's the poster boy for sudden cardiac death, SCD, from non-cardiac or non-antiarrhythmic drugs. So this drug showed no effect on APD or in vivo QT non-clinically as it was being developed. But once it hit the market, it was a big success, and cases of torsades de pointe, which were a big surprise actually, showed up. Now the frequency was very low, as Philip has alluded to. I mean, the frequency of these kind of events for non-antiarrhythmic drugs is on the order of 1 in a million, so you're never going to pick it up in a clinical trial. There was a lot of concern in this period historically for other drugs that were coming in with the same sort of thing, so the regulators switched over to, instead of concentrating on the event of significance, rare as it was, switched over to using the corrected QT interval as a surrogate. And so it's important to note that in the Seldane studies, at a commonly used dosage of 60 mg BID, the QTc was only increased about 6 milliseconds. That's a 2% increase. And so much of the heavy price that we've paid for impeding drug development has revolved around the very low threshold for QTc increases that evolved. And then, we were involved because we had shown in 1996, but we had certainly published in abstract form in previous years, that Hurg was the molecular target for terfenadine risk. So this figure does show the TDP record. Down below on the right-hand side, you see A and B, which is the Hurg current with the voltage steps shown on the inset, just to show you the terfenadine effects and how we measure the Hurg current. Importantly, underneath the figure is terfenadine and fexofenadine. So this tells you that there's no class action effects. By simply carboxylating two of the methyl groups on the end of terfenadine, which happens in one pass through the liver, you have a drug that's safe. This is Allegra, versus a drug relatively dangerous in this regard, terfenadine. So at this time, there were a bunch of non-antiarrhythmic drugs like estemazole, which is an antihistamine, cisapride, a prokinetic, droperidol, a tranquilizer, levomethadil, an opiate antagonist, grepafloxacin and sparfloxacin, which were fluoroquinolone antibiotics, and taurolidine, which is an anticholinergic. All of them popped up. All of them were withdrawn from the market for the very same reason. So during this sort of 7-year interval from 96 to 2003, there were drugs from a wide variety of classes. It almost seemed like there was an epidemic of this disease. So there are wide classes of drugs that have this propensity to block hERG, but there is no class action effect so far as we know, even to this day. So again, just the quick guidances. S7B came out in 2005, emphasized delayed repolarization, which it took the form of looking at non-clinically hERG, action potential duration, APD-90, generally APD-90 minus 30 sometimes, and ECG-QT. And then the TQT, the third clinical trial, you've heard about it from Philip, very low margin for safety here. And then CiPA with the change in emphasis that I've indicated. So in our own experience, we became very much aware of the fact that there were discordances between the hERG signal on, on the one hand, and arrhythmias and delayed repolarization on the other. So, for example, there was a publication by De Bruin and collaborators in 2005 in which they looked at a very, very large database, and they found that the odds ratio was 2 out of 3. That is to say, it got it wrong 30% of the time, at best, between a hERG signal and arrhythmias, which was the readout that they used. In our own experience, hERG and delayed repolarization also showed discordance. So if you look at the table there, you can see that we're looking at minus hERG, no hERG signal, plus hERG, a hERG signal, and then the columns are impaired repolarization and plus or minus. And so you can see that there's concordance roughly, in this case, 75% of the time, and discordance about 25% of the time. So these numbers of discordance running at about 30% or possibly even greater really point to the shortcoming approach, the shortcomings of the approach that have been used. And you've heard Philip talk already about the drugs that just don't fit in, like pentobarbital, amio. Verapamil in particular was important to me and to the way we went about it, because we knew all the way back in 1993 that verapamil was a potent IKR blocker as well as a calcium channel blocker. So we were very much aware of the contradictions here. Tolterodine, which shows up very much like verapamil. Ranolazine, you've heard about. Minoxidil is a drug that we have in the clinic that also has very interesting mice properties, which mitigate the hERG problem. So the results, I think Philip has talked mostly of this. I just want to mention one point, and that is that the FDA has a data set of 34 TQT studies, and there was 95% approval of drugs with 10 to 20 millisecond increases in QT, and 83% at greater than 20 milliseconds. So they don't even pay attention to their own boundary conditions. So the TQT is more honored in the breach than the observance, in my opinion. Okay, so our approach has been to look at multiple ion channels, and Philip has alluded to the 5 that he likes. In our initial approach, we actually looked at hERG, CAV 1.2, and NAV 1.5, but we are extending it to the other 2 channels. And what we did here, what's shown in this particular figure, is the results that we obtained. We looked at 55 drugs, and 32 of them we classified as torsatogenic, and 23 of them we classified as non-torsatogenic. And we looked at these 3 channels. Now, because we were looking at so many channels and we were accumulating so much data, these are usually 4 by 3, that is 12 data point IC50s. We had to do this in a sensitive system that had throughput, and we settled on the QPatch system for this, single hole. And what you see plotted here is the IC50s for hERG, CAV, and NAV. The data and the 95% confidence intervals are circumscribed by the circles that you see. And you can see that we were able to look at and get the IC50 data for these different channels. Now, how did we decide whether a drug was torsatogenic or not? We decided that based on the Redfern paper, on the CERT, University of Arizona database, and on the package inserts and the FDA adjudication of the package inserts. So we used all of these to weight whether the drug was torsatogenic or not. And then we were interested in the relationship to the safety margin. So we used Cmax and protein binding to estimate those numbers, or to correlate those kinds of numbers, so that we could then get a safety margin measurement. So the next slide shows you what really, I think, summarizes a lot of what we observed. So in panel A, you've got the ion channel IC50s, you've got the effective therapeutic concentration measured as by that asterisk, and then you've got the hERG IC50 by the diamond, the red diamond. NAV 1.5 is the rectangular blue, and CAV 1.2 is a circular blue fill. And so you can see that for terfenadine, the IC50 for hERG and the effective therapeutic plasma concentration are located near each other within an order of magnitude or so, but the other 2 are far, far away. On the other hand, for verapamil, you see that the IC50 for hERG and the IC50 for calcium channel block are virtually superimposable. And so when you plot the IC50 over ETPC, or safety margin, and you put the hERG as the takeoff point, you get these what we call V curves, where you draw the line that relates the CAV 1.2 safety margin to the hERG safety margin, and the sodium NAV 1.5 safety margin to the hERG safety margin. You get these V figures. Whereas with verapamil, which is such a potent hERG blocker, you don't get the V figure. You get, if anything, an inverted V figure. And so then if you plot these safety margin values against drug number, as I show here for plus TDP and minus TDP, the lines represent this 30 value of 30 as the ratio indicator dividing, that's the Redfern number for dividing torsatogenics from non-torsatogenics. We're a little more liberal, as the right-hand side of C shows. But anyway, you get the general picture. V's go with plus TDP, that is hERG's, where hERG is potently blocked, whereas the others are not. Whereas inverted V's go with potent hERG block, but with the block being offset almost always by very potent or almost equipotent block of calcium channels, CAV 1.2. So if you look at our data, you can model it, you know, since we were using a binary outcome in this particular index, either plus TDP or minus TDP, it works fairly well with logistic regression models, and that's what we applied in terms of modeling the data. So the left-hand panel, we had a bunch of models which took into account various combinations of hERG, CAV, and NAV. But the ones that we want to concentrate on are the one that looked at hERG alone and the one that looked at hERG plus CAV. And we looked at the raw data. We broke it down into decades of drugs. And if you see the plot under Model 1, you can see that there's not really very good there's a graded region right here where the separation gets a little blurred. On the other hand, if you use a model that takes into account the effects of calcium as well as hERG IC50s, and by the way, here you don't even need the plasma concentrations, although you need to keep in mind what they might be. You don't really need them for this kind of mathematical interpretation. You can see that there's a very good division between those drugs that are likely to be torsatogenic from those drugs that are not going to be torsatogenic because of the calcium channel offset, block of calcium channel offset of the hERG effect. So just to summarize this up, where things stood was 70%. False positives were most frequent, but false negatives occurred. Mice is better than hERG. You have to go to something to do this. It only becomes practical if you use automated gigaseal patch clamps. The mice model, number 5, was the best model we had. Some of the other models were okay, but this was clearly the best one for the moment. And it's important to understand that when you do it this way, you can get turnaround times that are very fast, much faster than manual patch clamp. So the cost compared to just doing the hERG assay alone is de minimis, especially when compared to the gain in predictivity. So let's talk about the future now and about CiPA. Just a few words. You've already heard what the deal is, and I think you can all see what the answer is here. You want to use multiple cardiac ion channels, as I've demonstrated. You want to use as a target something that reflects human cardiac electrophysiology, and that means stem cell-derived human cardiomyocytes. And we think it's very worthwhile to try to tie these together to produce in silico models of the action potential and of the ECG ultimately. And the focus should be on proarrhythmia, not on the QT. So here's how we've implemented this at CiPA at ChanTest at the moment. So I've told you about doing the mice business, and we do that routinely now. We also model this, as I indicated to you. We use those logistic regression models. This is simply a different display than the one I showed in the previous diagram. It displays the same sort of data, but in this receiver operating characteristic curve mode, where you plot the sensitivity. That is to say, you plot the positives against the specificity, or 1 minus specificity, which are the false positives. And you can see that if you use a mice model, if you use the hERG model alone, the price you pay is that to get the same sensitivity, you have to include many, many more false positives. Whereas if you use the mice model, you get rid of most of these false positives. So we also think that one should use the O'Hara-Rudy model to model the human cardiac action potential, and ultimately something of this sort to look at the QT. Now, for the integrated human cellular studies, we are using as target human embryonic stem cell-derived cardiomyocytes, either of the induced pluripotent type or from human embryonic stem cells. We're switching more and more to the iPSCs. This is the confirmatory data, and we're measuring this by manual patch clamp at the moment, and using the multi-electrode arrays where we look at field potential duration and arrhythmias. And we also use the Excelligence system, which is an impedance system, to measure cytotoxicity. And the idea then is to wind up with a proarrhythmia score, which will be mechanism-based, continuous scale, and rank-ordered comparisons. So over here on the right-hand side, you can see some of the data that we've been getting, and you can see when the arrhythmic events occur at multiples of concentration for drugs like ibutylide, these plus TDB drugs, thioridazine, droperidol, risperidone, taurodolene, moxie, sunitinib, amio, paroxetine, and the negatives like verapamil, metronidazole, tolterodine, and donepezil. So just to show you the MEA system that we use, you measure the FPD. This is now two-dimensional system. So what you're getting is two-dimensional propagation. And of course, as you know, repolarization is not a propagated phenomenon. So you get the field potential duration as indicated here, and you get the T wave here. Excelligence is an impedance measurement, which actually could tell you something about contractility as indicated over here. And just to show you, for example, data that we get in MEA with hERG block using moxifloxacin, which, as you know, is a positive control used for the TQT. And you can see that there is a strong dose dependence of the field potential duration, which is shown over here in the insert. This is baseline, this is 100 micromolar, and this is 300 micromolar. You see the QT getting longer, and here you see it whips off an ectopic beat. And what you see down here is that the spike amplitude actually gets smaller, which is because these cells, you know, they're not perfect, not by any means. They beat spontaneously, which indicates that they're depolarized. And because they're depolarized, you've got a certain amount of IKR activation or hERG activation. So when you put in a hERG blocker, they get even more depolarized, so the spike amplitude goes down. So little tricks like this that you have to be aware of as you proceed through all this. So anyway, the bottom line message is avoid unexpected outcomes unless you want to be a tourist attraction, as they are in Pisa, or you want to achieve expected outcomes, and I recommend that you visit, if you ever get a chance, the Geary Museum in Bilbao, Spain. Just like to wind up thanking my colleagues at ChanTest, Jim Kramer, Carlos Obejero Paz, Tony Lacerda, Yuri Kershev, Andrew Bruning-Wright, and a whole host of ChanTest staff scientists. Over at LeadScope, we've received help from Glenn Myatt, and from Ohio State University, statistical help from Joe Verducci. And with that, I think I will stop. Thanks.
Excellent, Buzz. This is Ernie Bush. We have really a whole raft of questions. We have roughly 10 or maybe 15 minutes left, so I'm going to jump right in. I want to thank both speakers, and then I'm just going to ask these questions, and please, one or both of you jump in and answer them quickly. But by far the first question is just my question. It's not actually from any of the audience, and it is if some of the members want to become involved with CiPA, how would they become involved?
They could certainly contact me. This is Philip, and kind of express, you know, how they'd like to become involved. There are a number of different working groups. They can also there's a lot of information actually about the CiPA efforts. And thank you for reminding me. I had meant to state this on the HESI website, and there's also a link on the CSRC website to the HESI website. But on the HESI website, there's a whole area that a lot of the key documents around this are being deposited. The idea is for this really to be developed in a very open, transparent manner. So you can contact me, you can contact Jennifer Pearson or Cyril Pettit at HESI, though Jennifer Pearson is the one who's kind of overall administratively providing a lot of assistance for this. And again, there are efforts, obviously, that are in the more clinical area with selecting compounds that should be tested and whether they've got clinical pro-arrhythmic potential and how much. And then there's in silico modeling and stem cells and ion channel efforts. Many companies are already involved in this, but certainly people could feel free to reach out to me.
Excellent. The next question I'll ask, and this comes up a lot, but the questioner says, how do we achieve consistency with stem cells, both from providers and across institutions. One of the big issues has been sort of a variance, particularly from vendors, but also different. So do you have a plan or a vision how we're going to achieve consistency on the stem cell studies across a wide variety of vendors and users?
Yeah, I mean, this is Buzz. This is a good question, and I mean, it gets to the. So it's early days, but I think we've made a lot of progress with stem cells. There are shortcomings. I agree. If the idea here is that I think there has to be agreed upon protocols for from the different vendors as well as from the end user of these, and we're certainly working on that. There is variability, and I think one is going to have to develop a fairly substantial database to sort through the various issues that are involved in variability and come to more standardized stereotype preparations. But we are doing that now, and we've certainly found with the iPSCs that we've been using that if you make sure that everybody is agreed about how the cells are going to be handled from the minute they arrive until you start to use them, and how many, how often or when they're going to be used after you started using them. If you standardize those methods and make sure that every time you get a change, that you can try to identify what the cause of that change or variability is, I think we will get this under control. There's no doubt about it that the data is getting better. Now, the other side of this is that right now, for manual patch clamp, of course, it's going to be laborious. I mean, there's no getting around it. We have, at ChanTest, we've got something like 4 or 5 manual patch clamp setups, and we're able to do current clamp action potentials on all of them. So we're hoping to accumulate data very quickly so that we can talk about the standardization at that level. Obviously, it would be great if we could automate this, but so far that has, because then we'd have much more data to play with, but that so far has escaped us. I do think the virtue of MEA is also the throughput there. And so, you know, you can often make up for the shortcomings of a particular, of some of these problems by just amassing large amounts of data and finding out, you know, where sorting through these data for where the particular variables might be impacting it. I know that's kind of a woolly answer to that, but that's where we're coming from.
Good. And I'll just jump right ahead. So there's another question. Actually, it's been asked a couple different ways. So people, I think, are very excited about the development of a model, in silico model for cardiac action potential. And the question is, will that model be public domain, or will it be generally available so anybody can have access, or will it maintain, be proprietary, and you have to go through a vendor to get access?
Well, there's a lot of this discussion ongoing with the in silico model and how that would actually happen. I mean, one approach is that people would be able to, you know, it would reside basically in the cloud, and people could input all their ion channel data into it and be able to, you know, get out the basically the results, so to speak. So I think the approach being taken is one towards open access. Exactly how that will happen and exactly what form, but for the in silico modeling, you know, I think the thing that we're all thinking about is that this is something that you know, it should be something that people can have access to and not have to pay to get their compounds run through it.
Excellent, thank you. And what are the timelines for CiPA to achieve its goals?
Yeah, that's a good question. I mean, as I understand it, Philip, you can jump in here too, that the idea, the goal at least, is for TQT to drift into the background in 2015, and for the proarrhythmia approach to take over, and then for the HERG assay, the S7B, will be revised in 2016.
Yeah, so the challenge that was laid down last July by the FDA was to have this in place in 2 years. So that will be a year from this July, and there's a lot of work going in that direction. It's there's a lot of work that needs to be done. It's obviously complex, but people are working very hard on it.
Very good. And a closely related question, which I think you more or less answered, but how do you, what is your plan, or how do you feel your chances of success of getting regulatory buy-in for the CiPA approach?
Well, the representatives to ICH are all part of the steering committee and all are involved. I think, in the end it's going to be based upon the data and the science, but I think there's a lot of interest worldwide in regulatory authorities about moving to a new approach that's more scientifically based, more efficient, and would prevent good compounds from not being developed. And I think they see this as a way to potentially improve public health. So I think there's, again, everything obviously is going to in the end be data driven, but I think there's a lot of excitement about it, and the ICH E14 group has also been informed, and there will be, in the future, more regulatory interactions.
Okay, it is the top of the hour. I think we'll go ahead and ask questions as long as we get them, but I did want to say thank you to the presenters and thank you to all of the attendees. There will be a full copy of this webinar available on the ChanTest website as the sponsors of this webinar, and you can go to their website and get a copy, which includes the slide deck and all of the audio recording. And I also wanted to say that there is a survey available in the chat section. We at CHA and ChanTest would really appreciate your feedback on today's webinar. So if you just take a minute to click on that link in the chat section, we would really appreciate it. With those two more or less commercial announcements, I will go back to a few more questions. I thought there was a question that's probably specifically for Buzz relating to the so-called mice assessment. One of the attendees has asked, how would ranolazine behave in the mice approach? And would it have been red flagged based on the limited number of ion channels used in mice? Is that question meaningful to you, Buzz? Meaningful to you?
Could you repeat the question, Ernie? I didn't get the second part of it. I got the first part, but would you give it to me once more?
Sorry, my voice is breaking up here. So how would ranolazine behave in the mice approach, and would it have been red flagged based on the limited number of ion channels used in mice?
Yeah, that's also a good question, an informed question. Right now, the way we've been doing it is we wouldn't have flagged it because we've been looking at peak sodium. We're very much aware of late sodium. We're trying to figure out, a way to do it. So if you're looking at the late sodium component of current, which is from the same channel, if you're looking at that late component, you would have picked up the ranolazine effect, and you could do it easily in manual patch clamp. Could you do it in Q-patch? That's what we're looking at right now.
Very good. Thank you. Another question. Is there a change in the ion channels listed for CiPA since last July? One of the attendees wants to know, are you expanding your list of ion channels, or is it pretty constant since the publication from last July?
It's pretty constant. There's been some recent discussion on kind of fine-tuning that, but I would say it hasn't changed significantly since the publication.
You know, here at ChanTest, we're looking at not just hERG, NAV, and CAV, but we are, and also looking not only at peak NAV, but also at late NAV. But we're also looking at, you know, IKS or KBLGT1-MIN-K or whatever the gene nomenclature for that one is. And we're looking at KIR, the inward rectifier. So those are the big five. But, you know, if there are discordances, there's nothing that prevents us, at least, from looking at every other possible candidate, and there's at least 13 of them in the heart that are major players. But you could go exhaustively through the list if you really wanted to track this down. So right now, from a practical point of view, probably the big three, I would say we should move towards the big five. And then, you know, for outliers, just go through the rest of them.
Yeah, so the focus at the moment is on IKR, IKS, the fast, and as well as the late inward sodium currents, the L-type calcium current, and IK1.
Thank you, and I will say again, for those of you that feel you need to leave, we do welcome your participation, and we understand we're past the hour, but I think we'll go ahead with these Q&As, and they will be on the recording, so people can still hear the rest of these questions. I
have to warn you that this is Philip Sager. I apologize. I thought we were just scheduled for the hour, so I may have to leave in a few minutes.
And the same applies to you, Philip. You know,
but I'm sure Buzz will be in really good position to answer.
Well, wait a minute. I've got a hard stop. I have a hard stop at 3:25. Okay.
Well, okay. So let's say we'll.
Well, it's only 12 o'clock in my time zone. Buzz, you've got three. Buzz, I know you've got 3 more hours.
Yes. I have a question here.
Go ahead.
Value added is, what will be the GLP status of the CiPA studies? Current ICH SMB are conducted under GLP. Will the CiPA work be conducted under GLP as well?
Well, for us, NAV and CAV are already GLP. Yeah, and we can make the other two GLP, but for the other ones, you know, I don't think it's practical at this point. It's not difficult for us. Well, not difficult. I mean, it's not difficult for me. For the QA people at ChanTest, it will be a problem. But, yeah, we can extend the SOPs that are necessary to the other two channels.
Yeah.
So
I think eventually the ion channels will have to be GLP. I don't think that's the in silico model is something that you know, is a little bit different because it's a software model. And then there'll have to be a lot of discussion and work regarding the stem cells.
Yeah, exactly. I think the stem cells, you know, you'd have to first of all qualify the vendors themselves, and whether they are or are not up to that, which would be, I think, not a GLP issue but a GMP issue. And once having done that, I mean, we can do the GLP here for them, and we can also, you know, if we got into the manufacturing end, because we are GMP qualified, we could do that as well.
So I have one more question here that I think is very good, and I think we've covered most of the rest of the questions, at least partially. So there is a question from a attendee, and the question is, can you comment on the importance of an in vitro assay that studies the actual contraction of cardiomyocytes instead of only the electrophysiology? So is there any interest or any consideration to look at functional performance as well as just electrophysiological performance?
Well, yeah, I mean, you know, first of all, the electrophysiology, though, Ernie, is really, that's the functional component of interest from an arrhythmia point of view, mainly. But nevertheless, I mean, you can use the Excelligence impedance systems to look at contractility, and as it turns out, they can pick up arrhythmias because the arrhythmias do manifest themselves in some cases as changes in contractility. So that is a reason. I think that's a reasonable backup. It's high throughput. It's not inexpensive. It has some really very important advantages at the moment, namely that you can, with that particular system, you can put the sheet of stem cells, the stem cell syncytia, in an incubator for days on end so that you can look at more chronic exposure effects. Now, you can't do that with MEA at the moment.
Very good, thank you. And I would like to ask everybody to click on the survey link and fill out the survey to help improve DSEC and feedback for the panelists. And with that said, I think we've covered all of the important questions. And I will ask Philip or Arthur if they have any closing comments, and then we'll close the webinar. Philip or Arthur?
This is Philip. I'd just like to really thank everyone for their attention, participation, the great questions, and it's really been a pleasure.
Yeah, this is Buzz, and yeah, I'll echo what Philip said. Thanks, everyone. Thanks, Ernie and CHA. I would like to just say that I think it's the present position in terms of understanding what's going on is untenable, and that CiPA is, at least at the moment, the most reasonable alternative and satisfactory way of resolving the issues, the discordances that we've talked about. Thanks very much.
Okay, thank you, everybody. And with that, I'll wish everybody a good afternoon, good evening. Bye-bye.